Good day. For today's topic is all about extracellular components and connections between cells. The plasma membrane is usually regarded as the boundary of living cell, but most cells synthesize and secrete materials that are extracellular or external to the plasma membrane. Although these materials and the structures they form are outside the cell, their study is important to cell biology because they are involved in the great many cellular functions. What is cell wall of the plants? Cell wall is an extracellular structure of the plant cells that distinguishes that them from animal cells. The wall protects the plant cells, maintains its shape, and prevents excessive uptake of water. On the level of the whole plant, the strong walls of specialized cells hold the plant up against the force of gravity. Prokaryotes, fungi, and some protists also have cell walls, but here we will focus on cell plants of plants. Plant cells walls are much thicker than plasma membrane, ranging from zero point 0.1 to several micrometers. The exact chemical composition of the wall varies from to species, and even from one cell type to another in the same plant, but the basic design of wall is consistent. Microfibrils, made of the polysaccharide cellulose, are synthesized by the enzyme called cellulose synthase and secreted to the extracellular space where they become embedded in a matrix of other polysaccharide and proteins. This combination of materials strong fiber in a ground substance or matrix in the same basic architectural design found in steel, reinforced concrete, and fiberglass. A young plant cell first secretes a relatively thin and flexible wall called the primary cell wall. Between primary cell walls of adjacent cells in the middle lamella, a thin layer rich in sticky polysaccharides called pectins. Pectin is used as thickening agent in jams and jellies. The middle lamella glues adjacent cells together. When the cell matures and stops growing, it strengthens its wall. Some plant cells do this simply by secreting hardening substances into the primary wall. Other cells add a secondary cell wall between the plasma membrane and the primary wall. The secondary wall, often deposited in several laminated layers, has a strong and durable matrix that affords the cell protection and support. Wood, for example, consistent mainly of secondary walls. Plant cell walls are usually perforated by channels between adjacent cells called plasmodesmata. In Figure 62, plant cells walls, the drawing shows several cells, each with a large vacuoles or nucleus and several chloroplasts and mitochondria. The transmission of electron micrograph shows the cell walls where two cells come together. The multilayered partition between plant cells consists of adjoining walls individually secreted by cells. The extracellular matrix or ECM of animal cells. Although animal cells lack walls similar to those of plant cells, they do have an elaborate extracellular matrix or ECM. The main ingredients of the ECM are glycoproteins and other carbohydrate containing molecules secreted by the cells. The most abundant glycoprotein in the ECM of most animal cells is collagen, which forms strong fibers outside the cell. In fact, collagen accounts for about 40% of the total protein in the human body. The collagen fibers are embedded in a network woven out of prote- proteoglycans secreted by cells. Proteoglycans molecule consists of a small core protein with many carbohydrate chain covalently attached so that it may be up to 95% carbohydrate. Large proteoglycan complexes can form when hundreds of proteoglycan molecules become non-covalently attached to a single long polysaccharide molecule. In Figure 63, extracellular matrix or ECM of an animal cell, the molecule composition and structure of the ECM vary from one cell type to another. In this example, three different types of ECM molecules are present: proteoglycans, collagen, and fibronectin. Some cells are attached to the ECM by ECM. Glycoproteins such as fibronectin, and other ECM proteins bind to cell surface receptor proteins called integrins that, did, that are built into a plasma membrane. Integrins span the membrane and bind on their cytoplasmic side to associated proteins attached to microfilaments of cytoskeleton. The name integrins is based on the word integrate. Integrins are the position to transmit signal between ECM and the cytoskeleton, and thus to integrate changes occurring outside and inside the cell. Cell junction. Cell is an animal or plant are organized into tissues, organs, and organ system. Neighboring cells often adhere, interact, and communicate via sites of direct physical contact. Plasmodesmata in plant cells. It might seem that non-living cell walls of plants would isolate plant cells from one another, but in fact, cell walls are perforated with plasmodesmata membrane-lined channel filled with cytoplasm. In Figure 64, plasmodesmata between plant cells, the cytoplasm in one plant cell is continuous with the cytoplasm of its neighbors via plasmodesmata. Cytoplasmic channels through the cell walls TEM. Cytosols passes through the plasmodesmata and join the internal chemical environments of adjacent cells. These connections unify most of the plants into one living continuum. The plasma membranes of adjacent cells line the channel of each plasmodesmata and thus are continuous. Water and small solutes can pass freely from cell to cell, and recent experiments have shown that in some circumstances, certain proteins and RNA molecules can also do this. The macromolecules transported to the neighboring cells appear to reach the plasdesmata by moving along fibers of the cytoskeleton. Tight junctions, desmosomes, the gap junctions, and animal cells. In animals, there are three main types of cells junctions. These are tight junctions, desmosomes, and gap junctions. All three types of cell junctions are especially common in epithelial tissues, which lines external and internal surfaces of the body. What is tight junctions? Tight junctions the plasma membrane of neighboring cells are very tightly pressed against each other, bound together by specific proteins, forming continuous seals around the cells. Tight junctions prevent leakage of extracellular fluid across of a layer of epithelial cells. For example, tight junctions between skin cells make us watertight by preventing leakage between cells in our sweat glands. Next, what is desmosomes? These are These function like rivets, fastening cells together into strong sheets. Intermediate filaments made of sturdy keratin proteins anchor desmosomes in the cytoplasm. Desmosomes attach muscle cells to each other in a muscle. Some muscle tears involve the rupture of desmosomes. Gap junctions. Gap junctions is also called communicating junctions, provided by cytoplasmic channels from one cell to an adjacent cell, and this way are similar in their function to the plasmodesmata in plants. Gap junctions consist of membrane proteins that surround in a pore through which ions, sugar, amino acids, and other small molecules may pass. Gap junctions are necessary from communication between cells in many types of tissues such as heart muscle and the animal embryos. That's all for today. I am Corazon Lim, your reporter. Thank you.